Caloric restriction, CR, defined as a sustained reduction in calorie intake without inducing malnutrition, has emerged as one of the most robust and consistently validated interventions for promoting longevity and delaying age-related decline across a wide range of species. From yeast and worms to rodents and primates, CR has demonstrated the remarkable ability to extend lifespan and preserve biological function, even under conditions that would otherwise promote metabolic deterioration. One of the most fascinating aspects of CR's impact lies in its apparent ability to preserve telomere length, the molecular clock embedded in our chromosomes that shortens with each cell division and acts as a key biomarker of cellular aging. The preservation of telomeres under caloric restriction appears to be intricately linked to a reduction in two major accelerators of cellular aging: oxidative stress and chronic inflammation. Both of these factors are strongly associated with accelerated telomere erosion, and both are significantly mitigated under caloric restriction. In animal models, CR leads to lower levels of circulating pro-inflammatory cytokines and reduced production of reactive oxygen species, especially within mitochondria. This more favorable biochemical environment decreases the burden placed on cellular repair mechanisms, allowing DNA, including telomeric DNA, to maintain its integrity for a longer period of time. Over the long term, this effect is associated not just with healthier cells, but with healthier tissues and organ systems overall. Perhaps one of the most encouraging aspects of CR is its systemic nature. Unlike interventions that target a single mechanism or tissue, CR influences multiple cellular pathways simultaneously: insulin sensitivity, autophagy, mitochondrial efficiency, sirtuin activity, and more, all of which intersect with telomere biology. This synergy may be why CR produces such consistently positive outcomes in studies. In mice genetically engineered to lack telomerase, and therefore experience accelerated telomere shortening, caloric restriction was shown to dramatically slow their rate of aging and delay the onset of age-related pathologies. In primates, long-term CR has been associated with improved cardiovascular health, lower cancer incidence, and preserved cognitive function, each of which may indirectly reflect the preservation of telomere length and quality. Beyond laboratory settings, emerging human data, though less comprehensive due to the ethical and logistical challenges of conducting decades-long dietary studies, also support the potential of CR to influence telomere biology in meaningful ways. Observational studies have found that individuals who practice long-term calorie reduction, particularly those who also adhere to diets high in antioxidants and micronutrients, tend to have longer telomeres and lower markers of cellular aging than their counterparts. These individuals often exhibit metabolic profiles similar to those of much younger people, with lower blood pressure, improved insulin sensitivity, and lower levels of systemic inflammation. While causation cannot be conclusively established from these data, the correlation adds weight to what animal studies have long confirmed. What makes caloric restriction particularly promising as a telomere-preserving intervention is that it does not require exotic therapies or complex genetic modifications. It is, at its core, a behavioral and dietary choice, one that could theoretically be adopted widely, with profound population-level implications. When implemented carefully, CR has the potential to not only slow aging but to enhance vitality, energy, and resistance to disease over the course of decades. Importantly, modern interpretations of CR emphasize flexibility and nutritional adequacy. The goal is not starvation or severe deprivation, but a gentle reduction in caloric intake that maintains all essential nutrients and supports sustainable health. The broader significance of CR also lies in its compatibility with other longevity-promoting strategies. It can work in synergy with physical activity, intermittent fasting, micronutrient optimization, and even certain pharmacological agents that mimic its effects, known as caloric restriction mimetics. Together, these strategies form a comprehensive approach to health span extension that is increasingly grounded in science and increasingly within reach. Ultimately, while caloric restriction is not a panacea, its effects on telomere preservation highlight a central truth of modern biogerontology: that aging is modifiable and that our choices, including how much we eat, can influence the pace at which our cells and bodies wear down. As more studies continue to unravel the precise molecular links between diet, metabolism, and telomere biology, caloric restriction remains one of the most hopeful and accessible strategies available for those who seek not just a longer life, but a healthier, more resilient one.